es el doctor Marco Cristancho from the Centro de Bioinformática y Biología Computacional (BIOS). He holds a bachelor's degree in microbiology from the University of Los Andes. He also holds a PhD in molecular biology of hybrid plants from the University of Manchester, and also holds a postdoctorate in bioinformatics from Cornell University. He has worked substantially in the resistance, modeling, and biomarkers generation of the coffee. As a general training, his experience is encompasses in the genetic improvement of the coffee. So I'm very proud to give place on stage to the Dr. Marco Cristancho.
Okay, thank you very much. Does everybody hear? Okay? Okay. Thanks to the organizers, to Janet and everybody who organized this wonderful course. So I come from this institute. I'm going to mention something about that. And I always criticize those speakers that start with an apology. But I have to start with an apology. I have to apologize because I do not have such wonderful pictures as Giorgio showed. And I'm not going to show such wonderful tools as Giorgio did. But I'm going to show, anyway, something exciting. It's DNA data. That's something that we, as bioinformaticians, manage. And we get really, really excited about managing data. So that's what I'm going to talk about today. And I'm going to talk about something that maybe Giorgio mentioned, and is the vast amounts of data that we have in different sciences. And sometimes we get so overwhelmed by the amount of data that we think is just so huge that we don't even think about how much is there. Something like astronomers started a few years ago to count the stars. Those are not infinite. It's a large number, but it's not infinite. And I came out reading a paper a few months ago of some guys who actually calculated how much DNA we have in nature. And we somehow can calculate how much DNA we have in a country like Colombia or anywhere in the world. So I'm going to talk about it. I'm going to concentrate very much in the studies we are carrying out here in Colombia. So let's start by talking about big data. Today, big data is an expression that is like common language in sciences. But when we review when this expression came to be like very popular, it's just a few years ago, not more than 5 years ago, that this expression became popular. Before that, we didn't talk about big data. It was something that, big data for biological sciences was talking about a large Excel file, talking about probably 1,000 data. But today, we know that big data is huge amounts of data. So in this paper, these guys mentioned about two sciences fields, astronomy and genomics, and two networks, two popular networks in the internet, YouTube and Twitter. Astronomy is the field in science that produces the largest amounts of data today. And we can talk about some expressions, some of them common. We know kilo, we know what mega means, giga, and from a few years ago to now, tera, because a normal computer today has about a tera of storage power. But these expressions, when we talk about this, peta, exa, zeta, and iota, those are kind of expressions that are not common for us. So I will say that from here, all this kind of data is big data. It's something that we cannot manage in a normal computer. We need servers, we need experts, we need special software, and even special operating systems. So all this is big data, and that's what we are managing today in several biological sciences fields. So for the year 2015, for the year 2025, we are expecting genomics to have more data than astronomy and to be the field that produces larger amounts of data than any other field in science. And one of the reasons is because we are expecting to have at least 1 billion people. I don't know what the population of the world is today. I believe it's 6 billion. But we're going to have about 1 billion of that population sequenced. The genome of those people will be sequenced. So it's likely that many people here in this room will have in their lifespan, will have their genome sequenced for some reason. Because you want it, because your doctor asked you to do that. So it's happening. It's actually happening. And the main reason that's happening is because of one particular technological development, and it's in a technology called sequencing. That sequencing technology developed in new technologies that we call today next generation sequencing at around this year, 2005. Before that, we just had one single technology, which was difficult to conduct, to perform, which was very expensive, and which produced little amounts of data, didn't produce vast amounts of data. But with these new technologies from this year, and we have now several technologies, we produce large amounts of data in terms of seconds. So it's something that is, it has revolutionized biological sciences, just one technology. And just as Giorgio mentioned some numbers, let's talk about some numbers that these guys in this paper mentioned. And we can calculate how much DNA we have in the biosphere, in the planet Earth. The amount of data that there is. I'm going to show you later how much of that is in Colombia. Well, actually, it's here in this table. So this table shows something about the most diverse countries in the world. And several countries here in Latin America, Colombia, Brazil, Peru, sequenced in France, and people from the States as well participated in that. And we are sequencing those two species, Arabica, which is a tetraploid, and eugenioides, which is a diploid. Sequencing and analyzing genomes of polyploids is very complex. It's much more complex than analyzing diploids. So in general, animals are easier than most plants to analyze at the genome level. Just to show you, we use some other techniques as well, apart from sequencing, of course. So in here, we show a picture of in situ fluorescent hybridization of coffee chromosomes, just showing that the Arabica, the modern Arabica, in fact come from these two species, and we can still recognize the chromosome that come from each of the parental species, which it shows that this is a modern hybridization phenomenon. So, as I mentioned, these are the organizations involved in that analysis. This is the genome size of the Arabica. We have completed that using this technology, Pacific Biosciences. So we have completed two complex genomes. Here are some of the data. We have assembled about 90 to 90%, 95% of the genome. Just to mention, even with the new technologies, it's hard to sequence a whole genome at 100%. Not even the human genome is at the 100% sequence. There are regions of the genome that are very hard to obtain the sequence. There are some structural reasons for that. There are some technical reasons for that. But we are close, anyway, to have a very complete genome. We are also working on orchids. Orchids is, again, talking about emblematic plants, is the national flower. We are doing this with Dr. Newbeck from Southern Illinois University. And he sequenced 14 species of orchids, some from Asia, some here from Colombia. And so far we have analyzed chloroplast and mitochondrial sequences, those that's been published. And we are trying to use the whole genomes to see how that changed the frame of these phylogenies. So that's something that is happening today, actually. We use something that are like biomarkers, molecular markers, some spatial sequences to do phylogenies, but the trend is to use whole genomes to do that. That's a huge task, but that's something that is happening right now. We are also studying some plants that are endemic here for Colombia. This is one of them. It's called chontaduro. We know it that is from the Pacific region, and it has uses not only in for the food industry, but also we know now that it has some chemicals that we can use for some industry, like cosmetics industry. So we are starting to analyze this plant. It comes, as I said, from the Pacific region, with some colleagues from the Universidad Nacional in Palmira. Plant pathogens. Plant pathogens is something that, because of my background, I have studied a lot. This is the first major paper that where a Colombian group was involved, and was the sequence of a Phytophthora pathogen that affects several plants. So a group from the Universidad de los Andes, led by Dr. Silvia Restrepo, was part of a large alliance in a large consortium several years ago that sequenced this Phytophthora, really devastating disease. And with the same group, and people from Senicafe, we sequenced the coffee rust, which is the major pathogen of coffee. In fact, it caused losses about 6-7 years ago all over the world, but especially here in Latin America. It can cause losses up to 30%. So it's a major problem. So we sequenced this pathogen, and it came up with some surprises. For example, fungi genomes are in general kind of small, some of them mural size, but this guy is huge. It has almost the genome size of a plant. We didn't know that before starting this work, so it was very complex to analyze. But we predicted all the whole set of proteins, and we also designed some markers for something that in these pathogens is called avirulence genes, which are the ones that are related to the actually causing the disease in plants. So these are some of the results. We published this a couple of years ago. And so we have about 22 secreted proteins which can work as biomarkers for the races of the pathogen. We are starting to work with some groups at CIAT, which is located in Palmira. This is a research center, international research center here in Colombia, that is dedicated mainly to 4 crops, cassava, rice, grass, and I forgot the other one, frijol. What's the name of that in English? Beans, that's right. But they also work with some other crops, like plantain. They also collaborate with several groups in doing different works in different crops. One of them, one of the analyses we are performing with them is a metagenomics analysis of a disease that is called cuero de sapo, which the guess is that it's a virus, but it's still not proved. So we are proving with these guys in CIAT several diseases of plants that we don't know was the actual causal agent. So these are powerful techniques. These new sequencing techniques are very powerful techniques to try to elucidate which are the causal agents of diseases in plants. And we are doing this for cassava. We are doing that for plantains as well. We are studying something like metagenomics of suppressive soils to a bacterial disease in plantains. We are also studying another disease of plantain that is called moco. We are sequencing actually that bacteria. And again, just as in cassava, trying to elucidate which is the causal agent of this disease, we are doing the same for plantain for a disease that is called elephantiasis, which today we don't know anything about the causal agent. We just know that it's a problem in plantains, but we have no guess of what is causing that elephantiasis. But we can make a guess, and we can accelerate the progress of identifying the pathogen by doing deep sequencing. This was done a short time ago by people in Corpo Ica, the study of Baculovirus that affects this insect, which is a major pathogen, a major pest, in maize here in Colombia. And today, this year, it has been started this project. It's a project called Colombia Bio. It's a project that is directed by Colciencias, but many groups in Colombia are participating already in this endeavor. So we are starting to study 2 groups of organisms in this first step of the project: insects and marine species. Those are the first goals to study. We know that, as I mentioned, it's a huge task to start studying plants, to start studying other microbes, to start studying animals. So we have to start from somewhere. And we are starting from studying insects and marine organisms. So these are some of the things that we want to do in this project. And the first one is something that is called bioexpeditions. That's going to be just the first step of the project. We are not just going to do expeditions of going to the ocean or going to the Amazon or going to the Chocó region and collecting material, because a lot of people have done that already. So we'll do that, but in a limited way, and we will study the collections that are already in herbariums, in live collections, in greenhouses all over Colombia. So bioexpeditions is something that is going to be the first part. So we participated in this one. It's called Expedition Seaflower Expedition. And it took place in this island, which is close to the island of San Andres, that belongs to Colombia. And in this particular project, there are a lot of different groups from all over Colombia and even from abroad. And we are in charge of this huge number of people. We are in charge of studying the metagenomics of biological communities of the reserve. So we have started to analyze metagenomics of corals and some other environments of that reserve. So some of the guys that we are studying is this guy, the Antelope bipinata. We are doing this with this group here. What happened? Something happened here. We are starting to study these marine species. Again, we are doing metagenomics, and we are sequencing some of those. That's not just to study biodiversity, but we know that those corals have biochemicals that are important, again, for industry. There are some dyes that are used for in the textile industry that comes from those organisms. Also for the cosmetics, again, we know that we can identify metabolites for that. For some of the species, we already know some metabolites that we have to look for in these studies. But for some of the species, they are completely unknown. So it's a challenge, but we are excited to take this challenge because something will come up of these studies that we are doing, again, with colleagues from Universidad de los Andes. Humans, and it's something that it sort of has struck me because I have worked mainly with plants in my life as a scientist. And I didn't know too much about what was going on in human research in Colombia and Latin America. I knew, of course, the Human Genome Project. I know all the human research that is going on in the States, Europe, Japan. But I didn't know too much if something was happening here. And the fact is that not much is happening in human research. And it's somewhat strange why it's not that happening here in Colombia. Just to mention, in most countries there are several projects to sequence large amounts of the population. Here, that's not happening. In most of Latin American countries, that's not happening. So we have to wonder why. But still, there are few things going on. So let's talk about this. Here in Colombia, there have been just two very small populations with some genomic studies. And one of them was in Medellin, population from Medellin, and another one was a population from Chocó. The population from Medellin was very narrow, in my opinion. It wasn't diverse. It was the same kind of people was sequenced. It was about 95 individuals, but all of them came out as expected, as almost the same genetically. And in this population, the ancestry is mainly European. And the other two ancestries are, of course, Native American and some African. But of course, in other projects where we studied population from Chocó, from the Pacific region, it came out completely different. The main ancestry of that population is from Africa. And with Dr. Kim Jordan from Georgia Tech, who is a colleague, he's actually going to give some talks here later in the year at the Universidad Javeriana in a diplomate that we are organizing here. He could identify 3 different origins of those populations from Chocó. And it was very interesting to know that because we didn't know that before. And also because some sociological studies were performed in this analysis. And just to mention, some musical instruments here are corresponding with these ones here, hundreds of years after these immigrations, which came, of course, because of slavery, the slavery of the colonization of Americans. We are working with a group here at Universidad Javeriana, and we are genotyping breast cancer genes in Colombian populations. we have actually come up with these genes that we are analyzing in a population. We are genotyping some human pathogens, like Vibrio. We are doing this with an institute called INVEMAR and the Instituto Nacional de Salud. We are also performing modeling of pharmaceuticals. This particular work was done with some colleagues from Universidad de Antioquia. They have developed several drugs to control leishmania, which is a tropical disease, and to predict which of those drugs will actually act over the pathogen. We do this protein docking, which is protein modeling, trying to predict that. So just to end up, I would like to mention something about my institute, the center I'm working on. And we are a very young institute. We have been working just for about 3 years now, but we already have presence in most of Colombia. We are doing some training courses because we know these bioinformatics and computational biology are new sciences in Colombia. So training is a very, very important way of taking this to many places. So we have diplomates all over Colombia with several of the best universities. In Bogotá, we are doing this here at the Universidad Javeriana. We started just last weekend. We do a lot of training courses in-house at BIOS in Manizales. And we have the collaboration of EMBL, EBI of Europe, Berkeley Lab of California, ICGEB. We have also had ICGEB courses in BIOS and some universities in Colombia. We are involved in master degrees, especially in Manizales. We have somehow also been involved in 2 master degrees in bioinformatics that are here in Bogotá. One is run by Universidad de los Andes and another by Universidad Nacional. So we participate in all those masters. This is our current cluster infrastructure. So it is a huge computer, and so far has worked well with major projects. Those projects I showed you, for example, about analyzing plant genomes, are huge projects. Plant genomes are big molecules, really, really big molecules, and are difficult to analyze and need big servers. So we have the computing capability of doing that. And next year we will triple this capacity with the project that we have with Chinese government. This is my team, my main bioinformatics team. We have even Romain Goudiot, who is from France, working today with us. And this is my main team today, doing analysis of data. This is the whole BIOS team in Manizales. We are right in the center of the country, right in the middle of the Andes, in the central coffee zone of Colombia. Thank you very much. I hope that I wasn't boring after Giorgio's talk. But we are really excited of working with so many people, as I showed you, here in Colombia, in other countries in Latin America. I didn't have the opportunity to show in the States, in Europe, analyzing these vast amounts of data. That's something that we believe in, that we think that a lot of development can be done in this country and everywhere by analyzing this data, by finding ways of controlling human diseases, plant diseases, a way of developing new chemicals for industry. So we are very much into this. Anyone who wants to work with us, just send us an email. We are willing to collaborate with anybody who has the needs of computing facilities, of expertise. We are here for that. Thank you very much, and continue enjoying your course.